Drug companies are generally most interested in developing drugs for common diseases. But one of the interesting and very powerful things about ASO technologies is the ease of development and the speed. Because these are informational molecules, if we know the sequence of a gene, we can very quickly design a steric blocker or a gapmer for that. And this has made it possible to develop these drugs for N of 1 diseases. These are diseases resulting from a unique mutation present in only a single individual. The first reported example of this approach to personalized ASO drug development involves a girl named Mila. At age 6, she was diagnosed with Batten disease, a fatal neurodegenerative condition. Whole genome sequencing identified a unique retroelement insertion that disrupted the splicing of the gene MFSD8. Now, the clinician who did the whole genome sequencing and identified this insertion had never been involved in ASO drug development before, but he had heard about the development of other splice regulatory steric blocking ASOs. And so he realized that this type of mutation might be amenable to ASO treatment. And so what he did was to design a panel of ASOs to the potential splice regulatory regions around this retroelement insertion using predictive algorithms that help in designing ASOs that could potentially correct splicing defects. And he compared two different chemistries, the 2'-O-methyl and 2'-methoxyethyl that we've talked about previously. And then simply using assays to determine the restoration of splicing in patient-derived fibroblast cell lines, he identified several oligos that restored splicing significantly. Toxicity studies were performed in cells and rats that showed these appeared to be safe, and with approval from the FDA, patient dosing was started in less than a year from the time that the mutation had been identified. There was early evidence of patient benefit when this was reported in the New England Journal of Medicine. She showed a reduction in seizure activity, but Mila's disease was already very advanced by the time treatment began. She already was blind and deaf and was no longer ambulatory. And perhaps because of that, she did die several years later. But this showed the potential for an individualized ASO to alter the course of a fatal disease. And it made many people realize that if an individualized ASO could be developed for Mila, there must be many other patients out there with unique mutations or perhaps mutations that only occur in very small numbers of people that could potentially benefit from having their own individualized ASO developed. And so the Oligonucleotide Therapeutics Society drafted a briefing document to try to provide some guidance to people who would be interested in this approach. And the FDA has issued several draft guidances to facilitate the development of N of 1 ASOs or individualized ASOs. And there are now more than 70 children who have ASOs in development to treat them, including gapmers. These are not all steric blockers. Now, of course, there's a high cost to developing these treatments. It was estimated that the cost of developing milasen was about $2 million. But we have to keep in mind that the annual cost of treating children who require ventilation and who are severely disabled can be over $400,000 a year. And so it can actually be cost saving if it's possible to develop ASO drugs that will allow those children to survive, let alone meet their normal developmental milestones. And so there's a lot of potential and interest in trying to accelerate this development. And of course, as more ASOs are developed, the cost and the rapidity of their development should go down, making this therapy more broadly available to other people. And this is a chart of the various steps in ASO development that we could include if there's time. It's just a schematic starting with the identification of patients who might be amenable to this, the genetic testing they need. This first phase takes a few months and costs relatively little. The second phase, where you actually get into screening the ASOs, takes longer. You need to generate specialized cell lines from the individual patient with their mutations, and that's very variable how long that part of it can take. And of course, there's no guarantee that you're going to come up with an ASO that will actually work to correct the mutation. And then the final phase is the most expensive. This is where toxicology studies are required. The FDA has allowed these clinical trials to proceed on the basis of only rodent studies. Usually ASOs require primates, but for this purpose, the FDA is trying to facilitate cheaper, faster ASO drug development.